Okay, this is the last name I'm going to kill, and she's from my group. Debarati
Roychowdhury.
Okay, there you go. sorry. Every single time. I cannot say it.
Good afternoon, everyone. I'm Debarati, and I'll be presenting our work on a text mining integrated knowledge map of microRNAs. And as you heard before, I've been working with Dr. Ariki for this project. So as we know, like microRNAs are non-coding small RNAs that regulate gene expression at the post-transcriptional level. And through this regulation, microRNAs can impact several cellular processes, and their dysregulation often lead to diseases. Now, we have a lot of resources with terms of microRNA. However, most of them cover different contents, such as target diseases and expression, and they're scattered across multiple resources. Some of them do not keep up with the most recent results, and a lot of them do not provide literature evidence, which is important for understanding the context of the result. So in this work, we are going to present an integrative text mining approach to collect microRNA information from the literature, capture all different kinds of biological entities present in these abstracts and their relationship with microRNAs. In this talk, I'm going to give you a brief overview of our resource, the different kinds of relations we focus on, the information that is stored in our database, the interface that we use to interact with our database, and I'm going to show you a use case using glioblastoma multiforme to show how we use the interface to interact with our database. The overview of our resulting resource is captured in this figure. First, we collect microRNA-related abstracts from Medline and store it in our database. Next, we mine the text from the abstracts to detect entities such as genes, diseases, biological processes, etc., and store it in our database, as well as we identify the relations between these biological entities and microRNA present in the text. So we store all this information into our database, and then we finally have an interface which can interact with this database, and the interface enables 3 different kinds of searches. One is the microRNA-centric search, where we get information specific to the microRNAs. The second is we can get microRNA-related information in the context of a specific disease. And the third is a keyword-centric search, where we can enter any kinds of keywords and in a general keyword, and then we can get microRNA-related information based on that keyword. And then we display our relations, and we have the ability to download the relations that we extract from all the abstracts. So now that I have spoken about relation between biological entities and microRNA, let us see what kind of relations we focus on. So recall that I mentioned in the first slide that microRNAs are essential gene regulators, and hence the first major relation that we want to extract are the relation between microRNAs and genes. Here I gave a small example to indicate that in this sentence, we would like to extract the relation between MIR338.5P and EFEMP1. Since the dysregulation of microRNAs often lead to diseases, the next major relation we would want to extract is between microRNAs and diseases. In this case, in this example, we would like to see that the high MIR93 levels lead to enhanced cell growth in hepatocellular carcinoma. In the case of diseases, we do a more fine-grained extraction. For example, we identify microRNA's impact on disease outcome or processes. For example, does it affect survival rate? Does it regulate the cell proliferation? We want to identify which microRNAs are upregulated or downregulated in the context of a disease. For example, whether in Alzheimer's, which microRNAs are upregulated or downregulated. Does the microRNA affect or impact the response to treatment of a disease? Do the authors feel that microRNA could be a therapeutic target? Are the microRNAs a biomarker? And in this context, it is important to know if they are circulatory microRNAs, and hence our interest in identifying their extracellular location. In our resource, we have employed several existing tools that have been developed in our lab. Mirtex is used to identify or extract the relationship between microRNAs and genes. Myriad was used to extract the relationship between microRNAs and diseases. Dexter identifies the microRNAs that are upregulated or downregulated in the context of a disease. And we built 2 additional tools that can extract the relation between microRNAs and their involvement in biological processes, and microRNAs and their location in the and whether they're found in the extracellular locations. So now that I've given you a brief overview of the different kinds of relation we are interested in, let us see what our database stores. So remember, we extract all the microRNA-related abstracts from Medline, but we store in our database, we store the abstract as a set of sentences in our database. We identify all the entities that were mentioned, entities meaning biological entities such as genes, diseases, biological process, extracellular locations, etc. We also store the spans of these particular entities from within each sentence. We normalize the IDs of the entities and store the normalization IDs. We identify the relation between these entities and microRNA. We also mention whether a particular relation is negated, and we store this information in our database. Finally, we assign the location of each sentence with respect to the abstract as to whether it's located in the introduction or in the results, thereby giving us an idea of how important a sentence is when we find that it has some relation existing between microRNAs and an entity. So this is like a pictorial representation of what our database contains. We take the abstracts, we stored all the abstracts from Medline into our database, and from there we take all the abstracts, we split it into sentences, we detect entities, we normalize them, and we find their relations. So for detecting our entities, we use publicly available NER tools such as PubDater to identify genes and diseases, and we build our own in-house NER tools to identify the mentions of microRNA, biological processes, and extracellular locations. We normalize these entities using the ontologies such as NCBI gene is used for genes. We use MIR-based to normalize microRNA mentions. We use disease ontology to normalize diseases. We use gene ontology to normalize biological processes. And we use Brenda to normalize the extracellular locations. Our relation detection tools, that is MIRTEX, Myriad, Dexter, and the additional tools use Stanford Core, Blip Parser and Stanford Core NLP in order to parse the sentences and generate their dependency structures. We extracted a total of 47,142 microRNA-related abstracts, and out of these, we found 32,696 contained some relation between the entities and microRNA. We keep track of all the mentioned pairs in the sentences, such as more than 800,000 microRNA gene pairs occurred in all abstracts. And among these, more than 50,000 were identified by MIRTEX. Similarly, we keep track of all the other mentioned pairs and relations between the other entities and microRNAs. So this sums up the information that we store in our databases. Now let us go see what our interface looks like so that we can interact with our database. In our interface, we provide 3 different kinds of search, as I mentioned earlier. The first one is microRNA-centric, the second one is disease-centric, and the third one is keyword-centric. So now if I want to look at a microRNA-centric search, we would provide the microRNA name in the search. Then that would give us a list of all entities that are involved or associated with this microRNA. And for example, if I click on proliferation, it's going to show us all the sentences. The next page is going to show us all the sentences where microRNA is involved in cell proliferation. In the second kind of search, which is the disease-centric search, we can either give the disease ontology ID, or we can type the name of the disease, and we have an auto-filtered list. That is, the results are provided by NCB or Bioportal. So because often we do not know the DOIDs of each disease, so it is easier for us to type the name. So here, if I type the name glioblastoma multiforme, it's going to take me to a page that shows, that gives me a summary of all the entities involved in the relation with the with the microRNAs that in the context of glioblastoma multiforme. So here in the bottom level, you can see all the abstracts where each of the microRNAs occurred along with the gene mentions, diseases, and biological processes that occurred with it. The table here, we can filter this table using the filter box. If we want to just focus on microRNA 7, we can enter microRNA 7, and the table is going to filter out and show me all the abstracts that have microRNA 7 and the other entities. If we want to narrow down our focus to individual relations, we can navigate through these tabs. And if, for example, if you want to see the gene regulation, I click on gene regulation, it takes me to a page where it shows me all the abstracts, the microRNA mentions, the genes that were targeted by these microRNAs, the kind of relation, that is direct, indirect relations that we have between microRNAs and genes. Again, here we are able to filter this table using a filter box. I want to mention that since each of these microRNA mentions and the gene mentions are normalized, clicking on any of them will take us, so for example, if I click on MIR30B, it is going to take me to the MIR-based page for MIR30. Similarly, if I click on PRRT2, it is going to take me to the NCBI page that contains information about PRRT2. If we are interested to explore the abstract, we can click on this abstract, and it's going to take me to a page like this, where we show each of the sentences in that abstract, as well as all the relations between the different mentions, different gene mentions, biological process mentions, and the microRNAs present in this abstract. So I'm going to show you, I'm going to further show you an interaction with the data, with our database using the interface through a use case. In our case, we chose glioblastoma multiforme because it is the most common malignancy of the central nervous system. And in order to identify the role of microRNAs in glioblastoma multiforme, we decided to follow this procedure where we picked a recent review of microRNA's role in GBM, and then we started exploring our system to see how much of the knowledge maps or information in this review we can get from our resource. So the review discussed 8 hallmarks of GBM and microRNA's role in them, but we focused on the first 2. So the first 2 were sustained cell proliferation via receptor tyrosine kinase signaling network, and the second one was evading growth suppressors. So if we come to the first question, which is about which microRNAs are aberrantly expressed that affect the RTK signaling networks in GBM, leading to sustaining cell proliferation, the answer to this question can be summarized from the figure that we, from this diagram, from the review that we took. And for example, if I want to see that whether I can identify all the microRNAs that inhibit PTEN, resulting in an increased cell proliferation, the way I will do it is using the second kind of search, which is the disease-centric search that I was just showing you 2 slides back. There I will put in glioblastoma multiforme or DOID 3068. I will get to the summary page of all the entities. And then here I can filter the table by saying I just want to concentrate on the abstracts that have P10 in them. So here I focused on only P10 since I want to see which microRNAs inhibit P10. And then I identify the following abstract that has proliferation in it. So I see, okay, MIR4943P is identified in this one, and MIR943P was one of the microRNAs that the review found to have inhibited P10. So we can further explore the abstract to see the textual evidence of the role of microRNA 4943P in cell proliferation in the context of GBM. But before I proceed to the abstract page, I would want to highlight on this particular MIR221, which we were able to extract from the different Medline abstracts, but this MIR221 was not identified in the review, but we were able to find an abstract that had MIR221, which inhibited P10 and was also involved in cell proliferation. So now let us see what the abstract page looks like. I've already shown you what the abstract page looks like. So if I want to explore the PMID 25662849, it brings me to this page where I see, okay, this particular row has MIR494 interacting with inhibiting P10, which is in sentence 10. And then I see there is one more sentence that says MIR494 3P is involved in proliferation, which is sentence number 0, which is actually our title. And this gives us a textual evidence of MIRNA 494 3P's role in inhibiting P10 and resulting in cell proliferation in case of GBM cells. Similarly, we did a similar exercise for the second question, which was about which microRNAs are aberrantly expressed that affect RTK. I'm sorry, I skipped a slide. So using this exercise, we were able to identify 25 of the 29 microRNA target pairs in the context of RTK signaling that were identified in the review abstract. We did a similar exercise for the second hallmark question, which was about which microRNAs contribute to GBM leading to evading growth suppressors. And we did a similar exercise. We went to the DOID page. We gave the different gene targets to see which are the microRNAs involved in it. And we were able to capture 26 of the 28 microRNA target in context of glioblastoma, where it was described as a microRNAs whose expression affects cell growth. Finally, in order to show you how we use the third kind of search in our interface, we decided to explore this question as to which microRNAs contribute to the resistance to temozolomide treatment, which is a drug that is used in chemotherapy. And for this one, we use the third kind of input search, which is the keyword-centric. Here we enter glioblastoma and TMZ or temozolomide treatment. We come to a page that gives us a summary of all the entities. Then we narrow down our search to disease response to treatment relation for that particular microRNAs. And here we see it gives us a list of PMIDs, the different microRNAs in those PMIDs, and that they affect or they have some effect on TMZ treatment. So we can explore one of the abstract pages. It's going to give us this abstract where we can see that this particular line talks about chemosensitivity of GBM stem cells to temozolomide, and we can immediately get to our textual evidence. So this brings me to the end of our presentation, where I gave you a brief description of what our database contains and how we can interact with the database. Users have different kinds of queries, and they can enter any query with respect to whatever their need is, whether they want microRNA-related information, whether they want some information in the context of a certain disease, and they can get an overview of they can get an overview of all the entities occurring in each of the abstracts, the relations occurring in each of the abstracts, and they can use this for literature survey or for any other purpose of research. And in future, we plan to extend our search and querying capabilities so that we can have multiple, we might want to search multiple microRNAs at the same time. We would like to integrate an external database to get a more extensive knowledge map for, specifically for whether microRNAs are biomarkers and which extracellular location they're located in. The first one of those kind that we would like to integrate, one of the external databases that is already present is Mirandola, and it contains it's a curated knowledge base of non-invasive biomarkers. It's publicly available, and it has and we want to integrate this so that we can have more extensive information about the extracellular locations. Our database will be automatically updated on a regular basis, and our interface will be publicly available soon at this particular URL. Currently, it is located on a local machine that is only accessible to the people of University of Delaware, and we are in the process of transferring it to our public server, and it will be available soon. But on that note, if anybody is interested to get a live demo of the interface, you can come to me after my talk. I'll be here for 5-10 minutes. I can show you how you can interact with the database, how you can interact through the interface. And on that note, I would like to end my presentation. Thank you, everyone, for listening to me, and I will take questions.
Time for one quick question. Anyone?
Very interesting work. I noticed that you mentioned that in both cases, you missed 2 by text mining compared to the review article.
Yes.
I'm just curious, have you looked at why those 2 cases were missed?
So we focused on the abstracts, and the mentions of microRNA for these cases were in the full text. So the reason we missed them is because we didn't explore the full text, but we plan to incorporate the full text as we go on in future. Hopefully, we'll be able to capture those as well.
We will resume at 2 p.m. So those who have presentations, please come before 2 p.m. to upload those presentations, or you can stay now and upload them. And see you soon. Thank you for attending this session.